CRISPR Cas9, I'm Layla, and
I'm Barbara.
how it's used. My video is video 3, which talks about how the CRISPR Cas9 was first discovered in bacteria, which are very diverse organisms. Specifically, the CRISPR Cas9 came from bacterial defense, which is when certain viruses attack bacteria and then the bacteria develops antiviral mechanisms to fight back from the virus.
My video was the 20th one, and the main points from the video is that CRISPR plays a big role in different areas of biology because of its versatility and programmability. The versatility of CRISPR creates more in-depth discoveries and questions related to CRISPR.
How it works.
Targeting. First is the targeting phase, where the Cas9 guide RNA is brought into the cell, where it is going to randomly associate and dissociate with DNA. Cas9 will then recognize and bind to a 3-nucleotide sequence called PAM.
After the targeting phase, the binding phase starts, which is when the Cas9 will recognize and bind to PAM in the cell's DNA. After the binding of the PAM, the Cas9 will unwind and pull apart the DNA double helix upstream of the PAM. If the sequence of the unpaired DNA strand is not a match, then the Cas9 will disengage from the DNA and zips back into a double helix. But if the sequences match, then the guide RNA base pairs and the complementary DNA sequence creates a DNA-RNA helix.
Cleaving. After binding the DNA, RNA pairing will trigger the Cas9 to change the 3-dimensional structure and will activate its nuclear activity. In order to do this, the Cas9 will split both DNA strands at the site upstream of PAM.
The last and final stage is DNA repair, where the CRISPR-induced double-strand DNA breaks can be repaired by either non-homologous end joining or homology-directed repair. This phase occurs because the cells contain enzymes that repair double-stranded DNA breaks, which can lead to mutations. Non-homologous end joining versus homology-directed repair.
NATJ vs HDR section. Non-homologous end joining are more frequently used, an area faster repair mechanism. This process involves the cells not using a template when joining broken DNA ends together. It is an error-prone process because it introduces mutations. Most of the time, these mutations are random.
Homology-directed repair is a less error-prone process that uses a homologous DNA template to accurately repair the break. By introducing the cell as an excess of DNA repair template along with the Cas9 guide RNA complex, it can easily be manipulated. This process allows scientists to change target DNA into a new sequence, as well as correcting an existing mutation.
One question that I have is, what specific diseases does CRISPR actually help?
One question that I still have is CRISPR completely safe to use, and what could be some of the side effects of CRISPR?